welcome everybody. We got a couple people in the room here, and I saw at least 12 or so online. I'm guessing that number is going to increase over the next few minutes. So welcome to the Tools and Technology seminar series. We are trying a new microphone technology today, so if people are having any trouble hearing, please mention in the chat box that that is the case. If you've got questions and you're online, you can put them through the chat. We'll be monitoring that, and we'll pass those on to the speaker. If you got a question in the room, raise your hand, and same thing, you can speak up and we can get those questions. So with that, I'm going to turn it over to our speaker, Jonathan Golob, and he's going to introduce himself.
Hello, my name is Jonathan Golob. I am an assistant professor in the Division of Infectious Disease. I'm a physician scientist. A little unusually, my background is in engineering, biomedical engineering and computer engineering. So my laboratory does host microbe interaction studies. And part of that, I develop and evaluate some of the software. So I'd like to talk with you about some of the things I've been working on to address a sticky problem. Just 1 second. Sure. I think many of you are going to be aware of microbiome science, this idea of trying to understand how the microbes that live within us and on us affect health and disease. This has really been a field that's exploded in the last couple decades, mostly facilitated by the advent of next generation sequencing, the ability to do large scale DNA sequencing, some innovative techniques, and then finally some software to handle the results of those analytic techniques. There's been a backlash over the last couple of years. One of the challenges in microbiome science has been one of generalizability and reproducibility of the findings. One can find many studies in the literature that find an association between some attribute of this microbial community that lives on us and within us and something, health, disease, treatment response. But even when the same condition is studied twice, it's been very difficult to end up with the same results. Likewise, even studies where there's an effort made to have a cohesive strategy, one central laboratory, there's been a consistent challenge in getting results repeated again. So this kind of falls more into the level of precision. The techniques have been a little bit imprecise, which has led to frustration among funders and critical evaluators of this research. I want to talk about the mainstay technique for microbiome research. By no means is this the only technique that's used in microbiome research, but it's probably the dominant one because it's efficient, it's tractable, and it works well. And that is to find a gene that is present universally in the microbes, in this case the bacteria, that has both highly conserved domains as well as highly variable domains. And use those variable domains as sort of barcodes to identify which microbes are present, and using the conserved domains that flank these variable domains as a primer site to amplify, to do PCR across the variable domain, amplify up those barcodes, and then sequence them. The gene that is used most often is the 16S rRNA gene. As a catalytic RNA, the conserved regions tend to be very conserved. Every base pair matters. There's no codon degeneracy like you would find in a protein coding gene. Conversely, there are some protein coding genes that are considered, things like CPN60, which is a heat shock protein gene. The 16S rRNA gene is not a requirement, but it's the most common used. And it has well-characterized variable domains. And for gut microbiome studies, V4, V5 have sort of become the favored region, but certainly there are other variable domains for use in other studies. For example, V1, 2, and 3 are favored among people studying the skin microbiome, as it's a little bit better, more entropy is contained in that variable region for the microbes that are typically found in the skin. How do you do this? Well, this is the same gene now, just schematized out flat. And so you pick the gene and you do PCR across at least one variable region, and you get a whole bunch of PCR amplicons. They go off to a sequencer, they get sequenced, and you get back essentially, depending on the technology, but at the end of the day, you get a long list of these reads, base pairs, if we're starting from the primer across the variable region and ending at the other primer. These then can be sorted or grouped or categorized into blocks based on the actual sequence identity. And then depending on what you want to do, you can go on to decorate these blocks of similar or identical sequences with things like taxonomy. This is this species versus that species. So this technique, it seems relatively straightforward, but like most things, the devil is in the details. How are the primers designed? How are they selected? What kind of polymerase is used? What sequencing technology is used? How big is this thing you're sequencing? Are you doing paired or reading it essentially twice, one from forward and one from reverse? And even just the software side of things, there's an enormous degree of variability. And the ideal approach here would give an accurate representation of the composition, structure, and similarity of microbial communities. What do I mean by this? So composition, it can be like there's this species is present and it's in this amount of dominance versus that one. And this often can be expressed in a matrix or a count table, which then can go into a lot of different statistical techniques that have been developed for that style of data. You can also look at a little bit more colligative measures of the entire community. Alpha diversity, which is a measure of both richness, how many distinctive microbes are there, and unevenness, or the relative proportion of microbes, is become a favored favored attribute of a microbial community to look at it. And then finally, even just in n-dimensional space, how similar are two microbial communities to each other in composition and structure? And there are a lot of different ways of doing that. And then finally, the thing that's kind of been lost is the generalizability. It would be nice if I develop a table on my data set, if it's meaningful to someone else working on their own population of patients, and could be immediately reused. How did I evaluate some of the tools? So when I started out as a postdoc, there's a lot of different ways to do this. I developed a package called Deckard that generates in silico data, which is this block I've just shown, that shares the composition of healthy human gut microbiota. But because it's in silico data, I know the origin of every single in silico read pair. And so I can go back and do the catalog. How accurate are different software packages at recovering and classifying these sequences? And if you just think about this from the perspective of trying to decorate with taxonomy, it can be correct, it can be undercalled, and that's not a bad thing. Sometimes there's just not enough entropy to really tell. And I would argue it's better for you say, hey, give me the species of this. And the software package says, the best I can do is family or genus level confidently, and not give me just an overconfident false answer. These are both desirable outcomes. You can also have something that's kind of a near miss, a miscalled sibling. So in this case, if the goal is species, you get the right genus, but you get called to a different member of that genus. And sometimes this is just the nature of taxonomy. As a clinical infectious disease doctor, taxonomists are my enemy. And so this is where I can get to give a little bit of shade towards my nemesis. And then you can have overtly miscalled. And what I mean by overtly miscalled is you're at least one full taxonomic rank off. It is genus A, and the classifier confidently calls it as a species in genus B. That is really undesirable. It's okay for things to be uncertain, but you don't want it to be overconfident. And then depending on the nature of the filtering and the overall approach, sometimes true reads from true specimens can disappear and be filtered out inappropriately by overly aggressive filtering techniques. So here's a bunch of different techniques that we evaluated using different approaches and different software. And you can get the rough sense of how many fell into each category. This is for species level classification. Some people would argue species level classification is not possible with this technique. And you can kind of see certainly some of the software packages will undercall, will say, look, it's in the right genus or family or whatever else, but not actually achieve correct species level identification. And so the thing that I'm often again looking at is the percentage of miscalled. I care about the blob. And then the other thing that I've always read about is when there's a lot of true data that is filtered out by the standard operating procedure of a particular software pipeline. Alpha diversity, like I mentioned again, Shannon is an integrated metric of both evenness and richness. How many things there are and how are they? This is true to true. And so it's a Spearman R squared of 1. Perfect correlation from true to true. here's all these different approaches, and you can see some of them do a remarkably good job of estimating this parameter, despite all of the challenges and complexities. You top out, you can see the bootstrap confidence interval for these. Some of these approach 95% R-squareds in Spearman. So again, we're looking for monotonicity, not necessarily true one-to-one, but because again, we often use these as a comparative metric. So I would argue to you, monotonicity or proportionality is really the objective, a reasonable objective here. So the scale might change, although a lot of times it's quite good. This is a much more estimatable parameter. So in the real world, why is this such a challenge? Well, getting back to my original premise, why has there been such a generalizability or reproducibility problem with this? Well, there's a lot of ways to get false amplicons. Taq polymerases are not perfect. I think it's become a standard to use error-correcting Taq, which does help. And there's studies that have demonstrated that helps on constructed communities. But it's a really true, Taq errors are really tricky to do. The polymerase is the most likely to run into trouble on the first round of amplification when it is facing template that is a lot of the non-standard bases and modified bases that occur in the actual underlying genome. And then once that mistake is made, it is fixed in each subsequent cycle of PCR and amplified up. So these false SNPs can look like real data and be really heavily represented in a specimen because of this early error that gets fixed. We worry a lot, historically have worried a lot about sequencing error. Sequencing technology has gotten very good. It's still a problem, and the sequencer also will give a sort of confidence based on the actual raw chemistry and fluorescence data, which is quite helpful and useful here. And then again, a lot of contemporary sequencing techniques will sequence the same amplicon twice, once from the fore, once from the reverse. And so you have that also as another fail-safe. And so compared to PCR errors, the Taq polymerase errors, it's a little bit easier to catch sequencing errors. There could just be primer dimers, chimeras, where there's a jump over. And then the really stickiest wicket when you're trying to compare from study to study is what exact primers are you picking? Even if everyone is like, all right, we're just going to work on V4, we're going to filter down to V4, there's no agreement, certainly in particularly in historic data, about what exact primers are the best primers. How degenerate are they? What is the exact amplicon that is used? And for a lot of techniques based on clustering, that's a real problem. You can try to trim and throw out legitimate entropy down to the common region, but even that's really tricky because indels and inserts do happen in these variable regions. And so it can be extremely challenging from a generalizability perspective. So let's talk about what people do. People will remove spoolable singletons. So you get one read of one amplicon, it's probably a sequence error. You can just filter that away, and that's the heuristic. You can likewise just a little generalize, say, this is a sequence variant that was only found in one out of hundreds of specimens we studied. Even if it's real, it's not going to be that useful as far as understanding the underlying biology. You can filter against a set. It can be a low stringent filter. You're not looking for very high identity or homology, but you can have sort of a catalog of what you think is right, and then you can use those. And then you can cluster at a percent identity. That kind of smooths out some of these incidental SNPs with only one base pair. You can also do this clustering on local sequence identity. I showed earlier on the software package Swarm that does that. And then you can use statistical modeling to deconvolute out error. So and this is some of the more recent packages that do this. So traditional techniques and a lot of published data is based upon sort of the combination of clustering at a percent identity just in the variable region, usually 97% identity, and then adding a filtering step against a reference of known good alleles. It's the low stringency. So I call this a semi-closed technique. You are creating these sort of clusters de novo from in software based on your empiric data, but then you kind of give them a little look over by saying, yeah, they got to look like something you've seen before in a global reference. Clustering does remove some true entropy in the spirit of getting rid of this erroneous entropy. And so it does worsen the experimental resolution of the study. And then again, this does absolutely nothing to solve the problem of primer selection. So on the basis of these sort of preliminary studies and evaluations and thinking through it, I assembled together in a workflow, a Nextflow-based workflow. I'm glad to explain a little bit more in detail what that is. A set of software I think that performs particularly well for this analysis and starts to systematically address some of these challenges. So Maliampi uses the combination of deconvoluting PCR and sequencing errors with a extant software package Data2. Part of that is to remove global singletons and do a little bit of filtering to look for things that just don't make a lot of sense. And then finally, there's this phylogenetic placement on the tree of known good full-length 16S rRNA alleles. And I will explain what this is a little bit. This is extant software that has been developed by the phylogeny people, computer scientists interested in phylogenetic analysis. But this is the magic that I think can start to solve some of the primer selection and generalizability problems. So what is Data2? Data2, you give the reads, you give all of the sequencers confidence of the reads, and you give it large blocks of reads that were cohesively processed in the same batch of physical chemicals. So I often want to say you have a 96-well plate where you're all doing the DNA extraction in the library. That is a batch. There's going to be that tack on that day, and that is thermocycler that had that, everything is as close as you can do. You have a set of paired controls with it. Data2 will make, based on the quality score, how confident the sequencer is, as well as where the base pair is from the 5' to 3' end of what was sequenced. And then we'll also start to think a little bit about the forward and reverse pairs, and does that, and so make a separate model for the forward reads and reverse reads, and then apply these statistical models to it. So there's a lot of variables to be fit, but the one thing we're not lacking in any of these experiments is a lot of data. There is always a lot of data. The order is on the order of probably tens of thousands of reads per specimen. And so even a modestly sized study of a couple dozen specimens, you're talking about in each read typically has on the order of 300 to 600 base pairs when you start to account for forward and reverse. So there's a lot of bases with a lot of data to fit. So this is, even though there's a fair number of variables you are fitting here, a prediction of mutation rate for every base pair based on quality score, for every position in each the forward and reverse reads, those number of variables is dwarfed by the sheer volume of data, often to the point where Data2 randomly will select reads to try to sample, but will not use all the reads because it's not necessary. It converges much more quickly. There's a nice review that I rather like that compares, there's multiple packages that do this. Data2, the reason why I selected Data2, and I confirmed some of these in my silico analysis, it finds more low abundance organisms without the cost of significantly increasing the number of non-reference hits. So it accomplishes both goals of filtering out gunk that was not actually there while preserving the low abundance taxa, where a lot of times there can be some real biology that's going on, and it's important to capture. That's why I picked Data2. There's certainly other software that can do it. This is just to get the first steps here. It gets rid of sequencing errors, gets rid of most PCR errors, most time errors. This still, as I mentioned earlier, cannot solve the problem of generalizability or primer selection. So let's talk about phylogenetic placement and what I mean by that. Well, the first thing I need is a tree to place on. And I use, to make my tree from references, a software package called RAXML. It is a likelihood maximization software. It is the bane of my existence. I am literally going broke running this. BT spins up a coal plant every single time I start running on this. Like any software, if it has MPI, you're in for a world of pain. So it's there, it works. There's a new implementation of the RXML NG that's supposed to be faster. I'm not so convinced on it, but it is a very beloved, really well-liked by people who do study phylogenetic science. And then once you have the tree, I have this placement business, and there's some really lovely places. P-Placer, which was developed by Frederick Mattson, who's now in Seattle at the Fred Hodge, and then has subsequently been re-implemented as EPA-NG, simplifies this problem down. This is actually quite fast once you have a tree that you can place it. So let's talk about that. Now, I'm going to go through step by step. I've been saying this multiple times. I'm going to explain what that comes off. You have your sequencer list. This is the output from Data2. I use it from Data2. This easily could be opticless output from Mother. This is agnostic. to how you get these sequence variants. These could be OTUs, these could be exact sequence variants, it doesn't matter. That stuff is independent. I like data too, but you can use swarm, you can use opticals, you can use whatever you want. And then you have a repository of full-length 6S alleles. What do I mean by full-length? Have to encompass the full gene, both conserved and variable domains, have no ambiguous bases. And then often if my end goal is taxonomy, I'd like to have them have a cohesive, concurrent, sort of by majority rules, taxonomic declaration, but that's not necessary. That's just gravy. If I really want to do it, I start with that subset. There's a separate software package that I have developed that honestly was first started at the Hutch by some of the lab medicine people there, that can call these directly from INT and filter them and bring them up. I am up to millions of full-length 6S RNA alleles, and just meet the sequence criteria. And then there are I think now approaching 100,000 that have actual proper full down to species, at least species taxonomic identification that is cohesive. Most of them are equal. That's fine. It's like. And so what do? Well, I search, just do a very low stringency search. My objective is to get at least one representative for every sequence variant. And then I filter these out just for, I can get an enormous number, even at like 90% identity or relatively stringent percent identities. So I do the top hit filter. Give me everything that matches at least as well as our best hit across the reference database, or maybe give a little bit of wiggle room for it. And then if I get a non-hit, I can go back and get some less annotated sequences depending on my objective. But what you end up is the recruits. These are full-length 6S rRNA allele recruits. Then I let RIXML take these recruits and assemble them into a de novo phylogeny. Other software packages do make phylogenies, but they make them on the sequence variant, which is just one variable region. This accounts for the entropy in all of the variable regions in the 6S rRNA allele, which may or may not agree with the bespoke variable region that was selected for a particular experiment. And this is important later on because they're full-length alleles. It's a lot less dependent on the primer selection. In fact, it's not at all dependent on the primer. The topography of the tree that is generated is not dependent at all. In a large experiment, this will be on the order of 10,000 reference sequences that will be into the tree. The goal is to have it be a little bit bushy, have a lot of leaves, a lot of branches, to have lots of bad choices to place actual sequence variants on so that you can be a little bit more confident the algorithms have had a chance to kind of nibble on less good ideas before it gets to the one that it should. And smaller experiments, this can be in the order of thousands. This is a little bit of a trickiness because the full-length 6S rRNA allele is about 1,200 base pairs. So there is a limit to how much true entropy is available to actually make the topography of the tree. So that's a limitation that I often struggle with, and that's why it's a trade-off between how complex the tree is and representative the tree is versus how tractable it is to assemble. Okay. And then the placement. So this is what pplacer, EPA, and G does. It uses, and I'm not going to do it justice, and I highly recommend you just read the papers if you are so inclined. But it essentially takes these variable regions, which are just a subset, and tries them out on all of the leaves of the tree, quickly settling down to subclades that seem likely, and eventually then gives you a likelihood for every node of the tree. Most of them are zero, so they don't mention those. Reports out any node of the tree, leaf or internal, where there's any likelihood that this particular sequence variant came from. Rarely does it just center on one leaf. Often the probability is distributed over a subclade. Rarely because of convergence. If you have a variable region, you can have a variable region here and here that look almost identical, even though overall they're quite different. So it'll dutifully apply the probability across multiple clades of the tree and report that out. And this is the data type that is used for everything else from here on out. And so it reflects the uncertainty. If you don't have a good leaf or good clade for your sequencer, it'll be at some distance from its closest leaf, which is another metric. So you get a weighted probability, you get a distance from each node in the tree, and you can go ahead and use that. And you can use this to calculate all the things we started off with: taxonomy, alpha diversity or just diversity structure of the community, composition of the community, and overall distance of how dissimilar or similar communities from one another. But you're reflecting the uncertainty all the way to the terminal analysis, which means you don't have to make so many harsh choices up front about, say, directly picking a reference or a narrow set of references to a site. Okay. It's a little bit tricky. I'm going to stop and make sure there are no questions on this point, and then I can send along here a little bit.
There are no questions so far in the chat box. If anybody has questions online, please feel free to enter them in the chat box or unmute yourself for now. That works too. Yeah, nothing so far. Thank you.
So how does this work in the real world? Let's apply it. I did some in silico validation, but I think it's much more interesting to talk about a hard real world application of this approach. So this is in collaboration with UCSF and the March of Dimes. It's a dream challenge. This is a machine learning AI challenge. The objective of the challenge is to have people be able to predict the date of collection of the specimen, as well as whether or not that pregnancy ended in preterm labor, based on the urogenital or vaginal microbiome. And what are we trying to do? We're trying to combine 9 studies with over 1,300 participants, divided over. And one of the objectives here is to have enough specimens and enough variance to start to get into some of these narrower questions about like distribution of how ethnicity and race affect some of these relationships. Preterm labor is obviously a horrible problem that we still are struggling to really understand well, and how to reduce the incidence of. And so, important problem, a real challenge to combine all these different data sets. They've been clearly heavily studied. Each study has a slightly different outcome that it has found. And so, can we successfully use something like an approach that I implemented in Maliambi to combine all these studies into a cohesive data set? Okay. So if we just generate sequence variants, these again are dotted 2 sequence variants. These are 6 studies for the sake of simplicity here. These are all new Metaanalysis 16S data. You can almost kind of see which studies kind of use similar primers. These 2, same basic primer design. These 2 have the same basic primer design. This one's different and this one's different. And so, sorry, these are ordination plots. These are Tisney plots. So the closer things are in this 2D space, the closer they are in n-dimensional space. I use cosine distance, which is actually a little bit more forgiving. I really like it as opposed to Euclidean distance, to sort of try to start to calculate it without having to do all this Phylogenetic placement. But you can clearly see these studies start to really segregate more by the study than by the biological characteristics. When was the specimen collected? And did this pregnancy end in term or not term, or what was the race or ethnicity of the person? This is after phylogenetic placement. And we've gone through all this business and then now recalculated distance between the communities based on the weight distribution on phylogenetic trees. And so there's a nice distance metric that has been developed, AR distance, which is a derivative of the weighted unifrac distance for people who care about it, but can account for this uncertainty of placement. And wow, same Tisney plots, the studies are far more overlapping. Now, it's totally fair for them to have a slightly different distribution of peaks in these contour plots, because some studies focus late in pregnancy, some studies focus early in pregnancy, some had more of different outcomes. But at the end of the day, the two-dimensional space in distance is far more overlapped and cohesive after this phylogenetic placement. Got it again. This is UMAP, another sort of ordination plot, same basic idea. These are 5 studies now where we have a little bit more metadata available. And so UMAP is just a different ordination plot. This you can kind of get the structure, but each study is a different color. And you can see they're nicely integrated. There's not like an island that is all yellow or an island that is all blue. And you can kind of see different studies contribute different specimens, but it's really hard to say any one of these is different from any of the other. This is, at least to my mind, very satisfying, cohesive integration of different studies with very different primer designs into each covering the same biologic site and roughly corresponding to the sort of setting. I just want to point out some of the, now I'm going to talk about structure. So that's overall distance, overall community distance and composition. Structure, or alpha diversity. It's really difficult. I feel like this is under considered how things like normalization techniques like rarefaction and primer selection and sequencing depth and all of the rest. Even though I showed earlier that a lot of programs can fairly well attribute it, it's a real challenge. I really like Dr. Willis's approach to this. I'm just going to make a shout out without going into too much detail about her essay and pointing out why it is important to think about the impact of variable sequencing depths and alpha diversity, but our go-to approach, which is very different than this phylogenetic placement business of rarefaction, makes things even worse. Okay, same story. Not normalized Shannon alpha diversity estimations just based on the sequence variance. And you can see, and they're now stratified by did the pregnancy end in term or not in each study. And you can see they each have a different baseline. They're all kind of jittered all over the place. And that fits again very consistently with different studies, different study designs, different sequencing and primer approaches, and sequencing depths. This is Shannon alpha diversity after this phylogenetic normalization. And here it's the baseline, the mode is almost identical in every single study. And then you can start to see again some higher alpha diversity in the preterm versus term that fits with some of the extant literature. Here's another one. You can start to see that pattern in at least those subpopulations. But again, it's much, much more cohesive. Two clear modes that start to be fit from study to study after this normalization. And then finally, I would like to have something for composition. And we could just do taxonomic assignment, and there's a way to do that. And the way the taxonomic assignment says is all of the weighted probability of the sequence variance is on the clade that is assigned to Gardnerella vaginalis. And so therefore it's got to be Gardnerella vaginalis. And then like I say, it's in the Gardnerella genus, but there's a little bit of some other ones all over it. It's probably vaginalis, but I'm just say Gardnerella. This is how key placer does in those sort of phylogenetic, how phylogenetic placement deals with taxonomy while still accounting for the uncertainty. As I've mentioned, taxonomy, particularly in some of the microbes I care about in the human gut, is a mess. You will have organisms reassigned from one complete phylum to another, basically at the highest level you can go from, aside from bacteria or not. Things get reassigned constantly. Taxonomy is flawed. And so what if we use the distance in phylogenetic space between sequence variants to really get at it? So one thing I want to point out to you is you have two different sequence variants, again, different primers, different studies, but from the same, biologically from the same underlying organism. They're going to end up in phylogenetic placement space, basically should be overlapping roughly into the same portion of the tree. So in phylogenetic space, even though in sequence space, these are going to be completely distinct from one another, they're going to converge and have a very short phylogenetic distance from one another. So you can go walk up the tree and walk back down the tree and get a phylogenetic distance between this group of sequence variants and that sequence variant. And you can use these paired wide distances to cluster with things like a kilometer cluster. And so you can get what I would call phylotopes. You group the sequence variants by their phylogenetic distance and setting others to cut off in this sort of distance space. Again, I made a software package that can do this. This is a Python-based command line utility. You can give it one of these placement files, the output from pplacer, EPN, and G, and you give it a desired phylogenetic distance, and it will give you the grouping at that phylogenetic distance. One thing I've been working on in the branch is to be able to do this iteratively, so you can add data in and it's reassigned. This is available like Maliampi as well. Maliampi works on Great Lakes and AWS and SPE and PBS Torque. I do a lot of collaboration. Everything I work on is portable and containerized. So it's implemented. You can try it out if you really get curious about it. How does it do? So pick the phylogenetic distance. So let's start over here at sequencer in space. We have each row is a study. We're at 6 studies again, just to keep this a little bit simple. This is detected, not detected. So each column here is a sequence variant. And you can see, yeah, again, W2 does a pretty darn good job in the studies that have similar primer design are kind of saying, yeah, these are the same. So there's a narrow set, but most of them are really completely non-overlap from study to study. This is the problem. Phylotypes. Phylotypes at a 0.5 phylogenetic distance, grouping these sequence variants, highly conserved across studies. And it's not unexpected that there's going to be some phylotypes are going to be one study or another. And when you start to consider the total read mix, how dominant are these in the treaty? These tend to be that ones deep in the tail. These are concurrently shared across all 6 studies, despite very different primer designs and sequencing of different centers and different studies. And so it starts to achieve one of our desirable principles, that we have things that more or less reference, I would argue, reference trees that are modestly dependent upon the topography of the full-length reference tree, but do not need an exact match of anything in the tree. And they're not dependent upon taxonomy, but can basically be turned into a count table. I ran through this quickly because I wanted to see if there's any discussion. I have another block that's going to talk about whole genomic sequencing, shotgun sequencing. If people would like to, I'm going to jump into that. But before I do, I'm going to pause again for some questions or thoughts on this particular 16S processing technique.
So far, there are no questions that have come in, but again, if anyone's online, feel free to put a question in the chat box or unmute yourself. Not seeing anything, and it also doesn't look like there are any questions in the room at the moment.
So one of the other proposed solutions to this problem is just ditch the primer selection. Sequence randomly primed DNA that's been acquired from a site. Sequence like there's no tomorrow. Sequence like Moore's law has not been killed by Illumina. Sequence till the end of time. Sequence like you're working on an HPC that is not cost recovered. And you get, because there are no primer selection, you're basically random primers. Everything should be hunky-dory and completely easily comparable. And what can you do with it? Well, you can get composition out of this, certainly, by aligning these random genomic reads. As you can imagine from host-associated microbiota, mostly what you get is, depending on the conditions, you can get a whole heck of a lot of human DNA, which is not as useful in this setting, so you have to throw that out. In disease states where there's a lot of cell death, that can be a lot of DNA that leaks out. That's fine, you can fix that. If you sequence enough, you can always make that better. Again, Moore's law is not dead, not fully killed. You can map these reads to reference databases. You can assemble them into contigs, which would be like little bits of genome in the world's worst and most horrible jigsaw puzzle. And then you can bin those together using these little pieces of jigsaw puzzles from different specimens to try to get genome-like material out of all of it. And that's what people do. And you have something you can associate with. It's a little bit harder to get estimates of alpha diversity, although it certainly can be done by that sort of like taxonomic assignment business. As you can imagine, random confounded with host that can be a variable percentage. You have even worse of a sort of limit of detection issues. And even in the study, comparability and having to think about things like, usually again, rarefaction and things like that can be problematic. Bacterial genomes are structured into little genomic islands that tend to be conserved in these clades of life and even be vertically or horizontally transmitted from organism to organism. So this is a set of genes that do something useful with their associated regulatory domains that do something the microbe needs or contributes a phenotype up to the microbe that is beneficial, deleterious, adaptive to an environment. The one my colleagues in ID fret about all the time are these little genomic islands, whether plasmid mediated or actually within a genome, that confer resistance to antimicrobials. Again, have the regulatory domain, often have multiple different antibiotics that they confer resistance, genes that confer resistance to multiple different antibiotics all together. So you get this horrible co-selection business. But when you start to think about the big picture objective, we want to associate microbial functional capability when we're talking about genomes with phenotype, health, disease, treatment response, preterm labor, it doesn't matter what. This is probably the minimal useful unit to start to think about. you could think about individual genes, but that is a lot of times genes really do need their other partners, and the microbes have already nicely assembled them into functional units to be able to control and regulate things. So why not try to identify these microbial genomic islands? Well, again, in collaboration with some of my dear colleagues, we developed a software package, Geneshop, that does exactly that, using a couple techniques to sort of make this a little bit more computationally tractable. A little bit more computationally tractable. Geneshot is horrifically computationally intense. If you're going to think about seriously doing this, apply for an exceed credit now. But regardless, here's what it does in the catalog. And if you excuse me, what I would do here. So you get the raw little itty bitty reads that have been randomly fine. Quality control filter on, chuck out all the human reads or mouse. If it's anything that is non-prokaryotic, you chuck it, and then you assemble into context. Okay. You take these contexts. So this is again the jigsaw puzzle part of this, and there's a bunch of different software packages that do this. None of them do it perfectly. That's fine. Life is all about working within precision. And then you can extract actual protein coding genes out of there. There's a bunch of software that can identify if something is likely to be a protein coding gene. And make a little catalog of them. You can deduplicate it. You can kind of try to figure out what the genes are by dumping them into annotation engines, but that's not required. And so you still have now a mapping that this centroid came from these genes were identified here. You do a little bit of deduplication to make it. And then you go back to the raw reads and align them against the centroids you've developed here. Why? In order to do the jigsaw puzzle, you need not just one representative read from that genomic region. You need a lot. You need a lot of layered up, piled up evidence. There, in a paper I'll show, but not go into great detail, what we found as a group is that when you're using these assembly-based techniques, you're trying to make the context, there's a limit of detection issue that varies by how predominant that particular genomic piece was in the underlying microbial community. If it's in the tail, it's going to be there if you really look for it, but it's not going to be assemblable by these algorithms because there just isn't enough overlapping data. So here, what we're taking, we're not looking at a specimen, we're looking at an entire set of specimens. So one of these specimens, if it's really relevant, it's going to be a little bit more dominant. We'll assemble it. And then you go back to the raw reads and you realign them and filter these alignments with a software package we developed to kind of handle multi-mapping reads. And then you can actually kind of flatten the limit of detection curve out quite a bit. Where if the read depth for that particular organism is anything above about 0.8, you can actually detect it. Whereas for contigs, you really need 4 or 5 copies of that genome to really start to pull out these contigs. Right. And so you end up with the big gigantic matrix of doom, gene sample count, and it is gigantic. We're talking probably in the order of tens of millions to hundreds of millions of genes in a decent sized set of specimens, hundreds of thousands of specimens. Disease states tend to be even more diffuse than health, and so this actually gets worse as you start to do it. So how do we dimensionality reduce a little bit more and find genomic islands? Why not look for co-abundance? Look for co-abundance across all our specimens. 2 things are on the same bit of DNA, they're going to co-vary. And so by looking for covariant groups, we can find bits of these genes that do. We can backfill from the information we had in the real world. We found these 2 genes next to each other from the context we generated earlier. But the co-abundant genes also can capture things that co-segregate because they're functionally selected to go. Microbe A creates favorable, or genomic island A creates favorable environmental conditions for genomic island B. They're going to co-vary even though they're different, maybe different organisms and different genomes with different mobile genetic elements. And so this is more of a conceptual, functional co-segregation in that set of disease or health. You can quantify those co-abundant gene groups, and that often reduces the dimensionality by an order of 1,000 or more. So you go from tens of millions to hundreds of thousands, which is still a bad problem, but less bad. You have a shot with power to do it, and you can kind of go through and annotate these co-abundant gene groups and so forth. And I've kind of gone through, this is this in silico software where we also talk about the dealing with the multi-mapping read problem, which I'm not going to go into tremendous detail, but is a real problem. Again, if you just take a read and you blast best fit it against a reference from one of these random, you're going to be wrong most of the time. Microbial proteins, like all proteins, all protein coding genes, are kind of made like Legos. And so if you have conserved domain, conserved domain, conserved domain, and a sequence from one conserved domain is going to match the sequence of one conserved domain of many other genes. And so this really needs to be thought of. And I'm just, in the spirit of time, I'm not going to belabor this, just to say our software package iteratively filters these out quite nicely to get down to the one true read, the one true source to remember read. Okay. Right, this is just a little bit, I'm going to also skip over this in the spirit of time. We apply Geneshop. Again, in a meta-analysis, this is another extremely hot area of incredible clinical importance. There's a new form of therapy for cancer that is now first-line therapy for over half of cancer diagnoses in the United States. Immune checkpoint inhibitor therapy. There's been some really lovely work that clearly associates functionally the structure and composition of the gut microbiome with whether or not the treatment works. Secondarily, the treatment can cause bad side effects, including colitis, and the microbiome seems related to that as well. What I mean with functional use of like adoptive transfer studies into mice, and you show the mice that also get an adoptively transferred tumor. You take a microbiome from a person whose tumor responded to this therapy, and you take a microbiome from a person whose tumor did not respond to the therapy. You put the same tumor into two different mice and give them the tumor microbiota. The mouse who got the responder's microbiome will respond, whereas the mouse that got the microbiota from the non-responder will not. So clearly involved, nobody can agree why or what. These studies all have a similar phenomenon, but cannot agree about why. What is the organism that is there? Each one of these senior authors will be glad to tell you which one it is, but they can't agree. And so let's try it. Let's pull them all together and let's see what we get. We ran it through Geneshop. My favorite kind of sequencing, someone else is paid for it. 108 specimens, billion reads, duplicate down to on the order of 1 million genes, further down to, so this is 7 million down to 1 million co-abundant genes. And then we do our regression against response or not. You get down to 3,000 co-abundant gene groups that seem to have any relationship to response. Not surprising, a lot of these co-abundant gene groups are core, not necessarily interesting. And then these, you can start to now do some annotation. One of the other big problems in metagenomics is the annotation databases are expensive and difficult to make and maintain, computationally expensive. Nobody wants to pay for them. So they get made once and then left to fester. I'm being a little bit too editorializing, but this is a problem, a challenge with this sort of data. So, right, we can at least get some sense of what they are, and we went through all this. Right. You can kind of get at least some sense of how well this works and that there is at least some degree of linearity. But the key thing I want to point out to you is there's just so much gene level diversity. You need to go through some of this business here. Okay. Let me show you a couple cases. This is the percentage between progression versus response, where this CAG was above the limit of detection. And then this is when it was above the limit of detection, how much of it was detected. So this CAG, and again, we're comparing two different studies. It's a little bit more compelling for this one, is positively associated with response. What is it? You look at the members of this gene. It's a CRISPR operon in F. presnitzan, which is an organism that is one of the few organisms universally present in the healthy human gut microbiome. How about this CAG? This CAG, it's the opposite relationship. Again, it's a little bit more driven by this study, but this is a CAG that is negatively associated with response. It's more abundant in people who would progress. What is this one? Well, member genes of this CAG are in a phage that is found same species, F. presnitzan. So, same organism, you would not be able to detect this in any sort of taxonomic-based identification. But if presnitzan has one island versus the other, and if you put these together, you basically do the ratio of the phage versus CRISPR CAG, you really start to get something that's quite discriminatory between outcomes, and start to understand that really you do need to start thinking down at these minimal units of genetic operons or genetic islands. So Geneshot is my answer to the question, well, can't we just use shotgun to do meta-analysis or get to learn a little bit about it? And so I'm going to stop there and just acknowledge all the amazing people that I get to work with and continue to work with, and the people who foolishly pay me to do some of this, not much. It's hard to get payment. I think that people know to do tool development and evaluation, but I'm endlessly grateful to the wonderful group of people I get to work with. So with that, I'll answer any questions. Otherwise, I very much appreciate your time and attention.